Thank you very much. Oh, thank you. Thanks. Well, thank you for that very, very kind introduction, Santiago. And this is very special for me because the first ever research talk I gave was to Art Winfrey. And it was when I was a first-year graduate student. And Jim Murray, who was my supervisor, so I was a student of Jim Murray, is how I get introduced, said, Art Winfrey's coming next Wednesday. So he always set it up so that his students would give talks to the visitors. So this put me in a dilemma because it was around this time of year. So the next Wednesday, I'd arranged with friends to go to Wimbledon to watch the tennis. So I had a choice then. Do I go to watch Wimbledon in the second week and watch some of the greatest tennis players on the planet? Or do I get the opportunity to meet Art Winfrey? So it was a no-brainer. So I gave a talk to Art Winfrey. And it was a terrible talk. It was a 10-minute talk on morphogenesis. And despite it being awful, he was so nice and supportive about the whole thing. And it really struck me then that here was someone who was so famous, so well known, but would take the time to sit there, listen to a first-year graduate student talk rubbish and be very supportive of them. And to me, that set in train that I learned a lot from his behavior. And I met him a few times since then and always had that same experience. So it's a great honor to get this award. And I must admit, when I did look down the list of previous recipients of this award, all of whom I am in awe of, all of whom I respect greatly, it doesn't seem right that I should be in the same list as them. But I'm very, very grateful for the Society for awarding me this honor. So I decided I'd split the talk up into three different sections. So the first section, I'm going to talk about cranial neural crest, cell migration, and try and show how mathematical modeling working with biology can lead to understanding and elucidation of new biology. And one thing to bear in mind is that mathematics can tell you what can't happen, what's impossible. It can tell you what could happen. It cannot tell you what does happen. That is what the experimentalists find out. Okay? I'm then going to talk a little bit about angiogenesis, and I picked that topic to show that even very simple conceptual models give rise to new mathematics and new open questions that we don't know how to answer. And then lastly, I'll talk about epithelial sheets, and some of this was mentioned earlier on today, and the idea here that there are different ways of modeling the same thing, and do those models give rise to the same result? And the answer is no. So I give this talk at Harriet Watt not so long ago, and I put this as a negative thing, that different mathematical models addressing the same problem gave different results. And Kevin Painter pointed out that this was the ultimate achievement in mathematical modeling, because biologists addressing the same problem with different techniques get different answers. And now we as mathematicians have done the same thing. So we've achieved the ultimate. We've captured biology. So I hadn't thought about that, and I thought, yeah, Kevin, that's really good. I must remember that. Okay, so we're going to talk about cranial neural crest cell migration. So what does that mean? So first of all, I must mention this is work with collaborators, experimental collaborators, Paul Klesa and Rebecca McLennan and their group, and then with Ruth Baker, who's sitting over there, my colleague in Oxford, and David Kay, and then with two graduate students, Louise Dyson, who's now faculty at Warwick, Linus Schumacher, who's a postdoc at Imperial, And those of you who were serial attendees of this conference will have probably heard these people talk about some of the things that I'm going to mention today. So basically, very early in development, cells, as you know, cells don't necessarily start where they're supposed to finish. They move to where they're supposed to spend the rest of their life. And so in early neural crest development, cells move away from the neural crest to the region in which they're going to do something. Okay? So the ones we're looking at are cranial neural crests, and they're the ones that are going to form this part of the embryo, of the adult. And so basically what you have is you've got cells that are sort of here, and they're having to migrate to here, to the branchial arches. And this is growing. The domain is growing. Okay? And as Mark Chaplin mentioned in his talk with the very nice quotes at the beginning, we have to simplify this. So we want to simplify this setup. Before we do that, just to say, well, this is important because if it doesn't occur properly, it can lead to developmental difficulties. Also, the link with health is that these behave very similarly to very aggressive cancers. So if we could understand how normal development occurs, then we might be able to understand how normal development doesn't occur in the case of cancer. So you can think of cancer as being a disease of developmental biology. And this is just to show you that with the advent of imaging and the huge advances over the last few years, that Paul can do amazing things. So he can track cells as they move along the neural crest. He can measure the length of this. This is in microns, so it goes from, roughly speaking, 300 to 1200. And this is stages, but roughly speaking, that's time in hours, roughly speaking. And so the domain is growing like this, and cells are coming out of the neural crest and are moving along there. Now, it's important to say that I don't have to say that to this audience, invasive cell motions have been studied all over the place. And here's a very nice example of looking at invasion of cells in the gut. And this is Matt Simpson, who's sitting over there with Kerry Landman and Don Newgreen. And this is something called Hirschsprung's disease, where if these cells are moving along a growing domain, which is your gut, if they don't reach the end of that, then you get this disease. And so they have a mathematical model, which I don't know if you can see this very well. This is actually for an experiment where there's two cell types going together. But basically, you've got a reaction diffusion equation. You've got diffusion. You've got proliferation as a logistic growth type thing. And then you can work out on that figure there under what conditions, under parameters, do you get full colonization or do you get the Hirschsprung's disease? And as well as doing a PDE description, they've done very nice cell-based, agent-based description as well. And so there's an example where you've got essentially proliferation is driving this motion. There's another method by which this occurs, and this is work by Roberto Maior's group and Karen Page, who was a former student of mine in Oxford, and Ian Cousin. And what they look at is this thing called contact inhibition co-attraction. So sort of idea there is that you basically got cells sitting here. This cell repels this one, and this one attracts this one. So then you get this sort of moving going like that. And so they have an agent-based model, a lot of mechanical interactions going on, and then you can see the sort of behaviors you get, whether you include these inhibitory and activation or attraction or don't include them. You see here you get the invasion occurring, and here A, B, and C, you don't get the invasion occurring. So there's lots of different methods that have been proposed for describing this. And in fact, that very computational work has been, and again, Mark Chaplin mentioned this earlier on today, This is work by Kevin Painter and mentioned Jonathan as well, and Alf Garish. And this is exactly the model that Mark showed, where you have this integral that gives rise to these non-local effects. And not only do you get this type of invasion of the neural crest, you also can get patterns to form. Pattern formation, stripes. So that's just to get the idea across that there have been lots of different models developed for different parts of the neural crest invasion. And this particular neural crest that we're looking at, it's not driven by division because no division occurs during the time that these cells move along. So the question is, how do these cells, basically if you think of it at an abstract level, and that's what a mathematical model is, we're going to take this curved growing domain and we're going to make it flat. The cells move very near, within 3 cell widths of the ectoderm. So we're going to crush down the Z direction and we're going to make it a 2D rectangle, okay? And it's going to be growing. So the 2D rectangle growing, cells come out at one end and they've got to get to the other end. What could be simpler, okay? And what we do is we use mathematical modeling as a hypothesis testing tool. So obviously, and we start with the simplest possible thing, hypothesis. When that doesn't work, we take the next simplest hypothesis and the next simplest and work up, okay? So first thing is, well, random diffusion. Well, random diffusion will take too long to get to the other end. So we need a signal. So why not have a source of chemoattractant and a sink of chemoattractant? That'll lead to a nice gradient. Cells move up that chemoattractant and they reach the far end. Okay? Problem with that is, if I have a gradient like this, it's too shallow. Cells won't be able to respond to it. Well, that's fine. I'll just move this up to here. And if I was a pure mathematician, that would be theorem done, paper published. But I'm an applied mathematician. I'm constrained by reality. So that quote that Mark put down about lovely theorem and ugly fact coming into the game. First of all, you can't have such a big source. Secondly, such a big source would make everything full of chemoattractant and the cells wouldn't be able to tell the direction. So that's not going to work. So what's the next simplest thing? Next simplest thing is that we actually know that the ectoderm produces VEGF, vascular endothelial growth factor. And we know that cells love to eat VEGF and love to move up the gradient of VEGF. So now you can see a model for that. The cells come out of the neural crest. They see the VEGF. They start to eat the VEGF. That creates a gradient. They move up the gradient, and they eating the VEGF, move up the gradient, etc., etc. And that is a very elegant and beautiful verbal model of a cell-induced invasion, but it doesn't work, as we see here. Okay? So what we have here is we have the region the cells are going to move along. We have this growing in the way that Paul measured. And then we have the chemoattractant being produced, diffusing, being internalized by the cells, being produced, and then you get this dilution term because of the growing domain. And we model the cells as entities, sort of little circles that come out here. They sample randomly the VEGF. They compare the non-local VEGF with the local VEGF. And if it's higher here than here, above a certain critical value, they move in that direction. Okay? Now, when you write down the PDE for this, what should the boundary conditions be? And I'll let you into a secret. I have no idea what the boundary conditions should be. So what we're going to do here is we're going to say that the VEGF is a sink at the boundary. And I'll come back to talk about the boundary condition a bit later on. So what you see here, the domain is growing. The cells come out. They start to invade, but they create a local gradient. So here, these cells don't see any gradient, so they don't move. So you just get some cells invading, and the rest get left behind. So this very nice model doesn't work. So now this is where we change from being technicians, where we simply took the verbal model, converted into mathematics, and see what the result is. And instead, we try to become scientists by making an hypothesis. And the hypothesis we make is that these cells at the back sense something, a cue from the cells at the front, and follow the cells at the front. What is that cue? We don't know. It could be that the cells at the back stick to cells at the front. It could be the cells at the front lay down a chemical cue, or it could be they lay down a physical cue. Again, going back to Mark's talk, where he showed the collagen alignment, where at one point you saw in his video the two cells are following each other. So that could be it. Our model cannot tell the difference between all those different things. It's too simple. So when we allow followers into the game, so now we have two different colors, yellow for leaders, white for followers, and we get the invasion. So that's the old one that didn't work. This is the new one that does work. One thing to point out here is that, look at these cells, they've got very far ahead. So they're not actually contributing to anything. These cells here, so you could imagine a situation arising where these cells might get too far ahead and leave everything behind. So I'll come back to that in a little while. So this model makes a prediction that there must be at least two different cell types. So Paul went and tested that prediction by looking at gene expression in a number of key genes from the front to the back, and found significant differences here in 19 of them. This is very old work. He's done a lot more since then. And there's clearly the cells at the front are different to the cells at the back. Okay, and that was work that was published 5 years ago. So what we did here then was we meet with our experimental colleagues and we decide on experiments that they can do in the computer, that they can do in the lab, we can do in the computer, and during that time, we don't talk to each other about science to make sure that we don't accidentally give each other a hint. And then we meet back 6 months later and we look at the results and we compare the two models. The animal model, which in this case is that's an important thing I should mention actually, is that when we talk about model, we usually mean mathematical model, but the experimentalists are also working with models. They're working with chick. I don't know about you, but when I look at a chick, I don't think, oh, human being. I say, oh, look, there's a chick, looks completely different to human being. So how come you do experiments in the chick if you want to find out something about the human? That's extremely abstract. It's almost as abstract as using a mathematical model to find out what goes on in a human. Okay, so we're all modelers. That's an important thing to bear in mind. So I'm not going to give you the details of some of these experiments. So they did things like they removed the trailing cells and the model made a prediction. That prediction held. Put leaders to the back. Model prediction agreed with experiments. I usually find that when the model predictions agrees with the experiment, you go, yes, and then you sort of think, well, so what? The really exciting thing is when they don't agree with the experiment, because then you know there's something more to find. That's when it gets really exciting. All models are wrong, but it's when they're wrong that they're fun, because then you can learn something. Okay? If your model is correct, there's nothing left for you to learn. Right? So they did the experiment where they put followers at the front. Okay, so they took cells from the back of a donor, stuck them on the front of the host, and here's the donor cells. Here's the normal cells here, and the model just doesn't give you the right answer. We tried to tweak the model, we just couldn't get it to agree. The reason is there appears to be phenotypic switching. So the leaders are changing to be followers. Oh, thanks, Santiago. The leaders are changing to be followers, and the followers are changing to be leaders. Okay? That's not in the model. So we've learned something. We've learned that these cell types are not ingrained. They change depending on environmental cues. Okay? So then that led to a new set of questions. And in this paper here, and then this was work of Linus Schumacher. So the first question, and for this question, you don't need a mathematical model to figure out what the answer is. What happens if you put some VEGF along the trail? Well, just think about it logically. What's going to happen? The leaders, if they're far away, they're following their VEGF. The followers, who are following the leaders, suddenly are in a position where there's lots of VEGF, and they switch their phenotype. They become leaders, and then they move up the VEGF. So that verbal model tells you that the stream should split into two. And that's what the mathematical model tells you as well, and that's what the experiment gives us. Cells moving along like this, here's the VEGF, and then they join up with the VEGF. In fact, some of the cells, the leaders that were moving towards that VEGF, turn around and come back to this VEGF. So the question Paul asked us was, how many leaders do you need? Okay? And this model can't answer that because we haven't parameterized this model with anywhere near the accuracy that's needed to give an answer to this question. But what we can do is give the qualitative answer of what happens if you change the number of leaders. Okay, so this was a new graduate student, Linus, so new color scheme, and new boundary conditions. Okay, so boundary conditions are now zero flux. See, I told you I don't know what the boundary conditions are. So we said, right, new graduate student, you can do zero flux boundary conditions. Zero flux for the important thing is whether we have zero flux for the VEGF or whether we have fixed to zero of the VEGF, qualitatively the results we're interested in do not change. So the results are robust to the boundary conditions. Okay? So here's the case where you've all leaders and you reach the end. Here's the case where 1 in 20 are leaders, you reach the end. So we said to Paul, it doesn't matter how many leaders you have, you will always invade. But look here, not very many cells made it, whereas here, lots of cells made it. So what we said was, the fewer the leaders, the better the invasion. Or put another way, if you increase the leaders, you would see worse invasion. And that's represented here. This is distance along the track. This is the wild type, normal. And this is when there's more leaders in the model. You still invade, but a lot fewer. Now, this experiment they can do. They can give something called HAN2, which upregulates the leader phenotype. And here's normal, control. And here's HAN2, more leaders. You still reach the end, but there's a lot less of them. Notice these scales don't match, but we don't expect them to match because we're not doing this quantitatively. Okay, so now let's come to the boundary condition. Okay? Now, Ruth Baker will enjoy this because this is my attempt at animation, Ruth. Okay? So remember the cells of cell track going like that, cell track going like that, cell track going like that. And so we said, and we put in all these different types of boundary conditions to stop the cells from going like this. And we said to Paul, there must be something here that's inhibiting the cells and down here. And beginning of last year, he said, you're right. I found this thing called DAN, which is a BMP inhibitor, and it's present there, and it's present there exactly as you said. We were delighted until he said, it's also present here. Right, so that was a piece of information we could have done without. So we thought, oh, fantastic. So now the cells are inhibited, and they still go. And here's just some pictures to show that, so I'll not go into that since we're running out of time. But here you see, this is the control. This is when you add DAN. And then it's a BMP inhibitor, so if you add BMP, it goes back up. And so, of course, remember I told you right at the beginning, these cells behave like melanoma cells. So what's the obvious next step? Confront melanoma cells with DAN. And if you do, they don't invade. The invasion goes down. Okay, so we've learned something about cancer. So what we think is happening now, remember I told you right at the start that we had to be careful in case the leader cells didn't get too far ahead. Now what we think is happening is that DAN is restricting the speed of the cells. So this is simulation that Linus did, where we have VEGF being produced along here, but it can diffuse everywhere, and cells come out of here, and this is where the DAN is present and reduces the cell speed. Here it doesn't reduce it by very much, and you get these leaders getting too far ahead, not signaling into the followers. Here it restricts it too much, and the cells don't invade. And here the cells are invading, and they're also linking to the leaders, to the followers, to signal for them to move. So we think that what DAN is doing is it's controlling and making sure that the followers, that the leaders don't go too far ahead. Okay? It's sort of taxing the rich to help the poor. Okay, so remember we started off with what could be simpler. I've got a bunch of cells here. I have to move them over to there. That should take a few minutes. What we find out is that you need to have at least 2 different cell types, leaders and followers. In fact, it turns out there's many more than that. There's phenotypic switching that depends on the environment, and we don't quite know how that works at the minute. We have a reduced cell velocity to make sure the leaders don't get too far away from the followers. And it's important to point out that this phenomenon of leaders and followers is not uncommon. It occurs all over the life sciences. Animal movement, tumor dynamics, social sciences, etc. And that leads me to the next thing I'm going to talk about, which is angiogenesis. So angiogenesis is also an example where you have leaders and followers, but it's to do with tumors. So the previous work was to do with normal cell movement, and this is to do with pathological cell movement. And this is work with Helen Byrne, who many of you know, and Samara Pillay, and those of you who were at the session earlier on today would have seen the very nice talk that Samara gave. And I'm not going to repeat her talk. I'm just going to point out some aspects of what she said. So in wound healing and in cancer growth, if you have a deep wound, your blood vessels are damaged, new blood vessels need to form, and that's done by the process of angiogenesis. A growth factor in fact, happens to be an isoform of VEGF. So VEGF crops up again. And VEGF is secreted either within the wound or by a tumor. It diffuses. Cells from the intact blood vessels start moving up the gradient of VEGF. And behind these cells, which are called tip cells, you get follower cells, sprout cells, that divide and move along behind. And just like in the neural crest, tip and stalk cells can switch phenotype. Okay, so this is a multi-scale problem because you've got things going on inside the cell, and then that's leading, and then you've got signals, oxygen and VEGF, and then you've got things moving along the blood vessel. And there's been a huge amount of work done on angiogenesis. I'll not bother showing the movie, but you can. This is stuff that a postdoc, Holger Perfel, did with us a few years ago, Helen Byrne and me. And you've got these blood vessels here, and then they do angiogenesis, and then you see here, this is the normal cells, and then this is tumor cells, and then you get this big tumor developing. And then you can do things like you can take an actual vasculature and encode that in and do things like this. And there are lots of people in the room, Roland, Tim Seacombe, James, various other people who've done a huge amount of work on angiogenesis and all these different networks, and then you put nutrients along those different networks. I'm not going to talk about that. The point about this is modeling the conceptual model that was actually proposed many years ago called the snail trail model. And so the idea here is that here's the tumor, produces VEGF, here's the blood vessel, and the tip cells, they go, as they move along, they leave the trail of followers behind them, and that's called the snail trail model. Okay? And this goes back to Leah Edelstein-Keshet talking about fungal growth, and Balding and McElwain, and Byrne and Chaplin. And the idea here is if you let N be the tip cell density and E be the endothelial sprout density, then in one dimension, this is the flux term, movement up the gradient. This is branching occurring due to the cell, the chemoattractant. And this is when a tip cell goes and hits a vessel, it gets annihilated and forms a loop. That's a negative. And then the last term is when two tip cells come together. Okay, you get that thing there. And then the idea here is this is the gradient of the flux. And the flux, the movement, is what's causing the trail to be left behind. So that's this term here. Okay. So the whole idea of this is to say, I've got my cells moving along, they're seeing a gradient, they're moving up the gradient, and they're leaving behind a trail. And that's the model that we all use. Okay. Now, there have been several examples in the literature where people have taken this model and discretized it to get pictures like this. And I do believe this is the most highly cited paper in the Bulletin of Mathematical Biology. So I agree with Mark. The Anderson-Chaplin paper, seminal paper in this field, and that's taking the continuum and going to discrete. And so what Samara did was say, what happens if we go discrete to continuum? So in other words, take that verbal model we mentioned of cells moving along and leaving behind a track, write a discrete version of that, expand everything, take limits, and get a PDE. And the PDE you get, here's the Byrne and Chaplin model, and here's one of the PDEs we get when we ignore volume exclusion. And you see they're both quite different, right? This is very challenging mathematically. This is linear. And even though it's challenging mathematically, you can do stuff with this. You can analyze this. You can work out wave speed, et cetera, et cetera. How do these compare? And to do these, we had to make a mean field approximation, which was made down here as well. So we now have three models. The cellular automaton, which is a true description of the snail trail. The Byrne and Chaplin model, the one that we all use, and the new continuum model that Samara came up with. So let's compare them under different circumstances. And you probably can't see here, but there are three lines here, and they're all on top of each other. So the models all agree with each other in this case, where we're assuming there's no anastomosis. In this case here, where there's tip-to-tip anastomosis, again, they all agree. So a mathematical question is, and I don't know the answer to this, how come these two equations, which are very, very different from each other, seem to behave in the same way? It's quite amazing. So if you're given the choice of solving this one or this one, you're always going to solve that one because it's simpler. Okay, so here they both work, here they both work, and here they both PDE models again give exactly the same answer, and they're both wrong because the answer should be just 0. So these two models that are very different mathematically give rise in a lot of the parameter space to the same answer. That's a puzzle, so that's an open question. What Samara found from doing this in this very systematic way is under what conditions does this thing break down? And she's been able to come up with key results like you have to worry about the cell densities. If the cell densities get too large, you get a negative diffusion and the PDEs don't behave properly. And where the models break down is when the mean field assumption is no longer valid. And Samara's talk was to look at the effects of volume exclusion. Okay? So once you put volume exclusion into it, the role of volume exclusion processes leads to absolute chaos. Okay? And, well, it doesn't quite, but I couldn't resist saying that. Okay? It doesn't lead to absolute chaos. What happens is Samara's done very nice work of showing under what conditions, so when you do volume exclusion, you get a set of PDEs that are even more complicated than the ones I just showed you. And then you can see, and she's done a lot of simulations on this, under what conditions do the simpler models break down. And the eventual goal of that work is that can we look at a real network, do some statistics on that network in terms of number of loops, densities, loop length, etc., and then be able to figure out, have a lookup table that tells us which is the simplest PDE model that can capture that. So that's what we're trying to do at the moment. And so the last thing I want to talk about is the last 5 minutes. And again, this has been mentioned. Mark mentioned this a little bit, and James Osborne in the previous session mentioned this. And this is work actually with James, Alex, Fletcher, Jupit Francis, and David Gavigan did all this work and published quite recently. And they used the chaste computational framework. And there are lots of computational frameworks around, and many people in the room have developed these different computational frameworks. And what we did in this paper was take 5 of the well-known cell-based models and ask them to solve some simple questions. Proliferation, adhesion, short and long-range signaling. Now, these models do not capture all the models that are around. It's just a selection of them. So the cellular automaton, where you just specify rules, you're on some sort of lattice with a certain rule, you move to different places, etc., etc. Cellular POTS model developed by James Glaser, which essentially boils down to you write down a Hamiltonian and you try to minimize a Hamiltonian, overlapping spheres, Voronoi tessellation, vertex model, all essentially can either be viewed the way Mark said of writing down Newton's law, or can be viewed in terms of writing down a Hamiltonian and then minimizing it. And so all the physics and all the modeling goes into writing down the Hamiltonian. I'm not going to go through that. Instead, I'm just going to say what happens when you now use these models to answer the same question. So the first question is, there's two cell types. These are classic experiments done by Steinberg in the 70s and 80s. You take cells from different organs, you mix them together, and they sort out, because they have different adhesion properties. So what we do here is we have our 5 different models, and there's 2 cell types here with different adhesive properties, and they should sort out so that you get the greens together surrounded by the purples, and then we just run. When I say we, I mean James and Alex ran these, and you see what happens. Even though this is wrong, this is my favorite, because it sort of looks like an upside-down teddy bear. So I quite like that one. Okay? So same problem, different solutions. We've eventually become biologists. Monoclonal conversion in the colonic crypt. So this is where in the colon, due to neutral drift, the colon converts. These little crypts are little organs present in the intestines, and they all convert to be one type of cell after a little while. And you see here all one type, all one type, all one type, all one type. These all agree with each other. This one doesn't quite agree. This is an example where we have short-range signaling, so notch delta. So we have little ODEs to describe notch delta signaling and how they interact. It's a sort of cell-to-cell interaction. And then we plot the expression of delta. How do these different models predict the expression of delta? And you see here, roughly speaking, they're all the same, but there's still differences between them. And then this last one is where there's a Morphogen DPP produced along the midline of the Drosophila disc. It's diffusing outwards, and the cells are growing in response to that. And what we see here is, roughly speaking, every model gives you the same result. Okay? So we have a combination here of sometimes the models all agree, so it doesn't matter which model you use, and in other times, the result that you get depends on the model that you use. And then this is a little lookup table summary of under what conditions, for what problem, which sort of approach is highly appropriate. Okay? But this is just the start. You can see that this is a very challenging problem. And as we get more into looking at individual cell-based models, we need to be absolutely sure that the results that we get are the results of the biology we've put into the model and not an artifact of the model we're using. And the example I like to give of this is that if a biologist came to me and said, I see an oscillation in a chemical, and I think it's caused by diffusion. I would write down the diffusion equation, I'd simulate it, and I'd get oscillations, because I don't know how to simulate. But there's a theory that tells us that if you've got diffusion, you will not get oscillations. And there's a theory that tells us that delta t over delta x squared has to be less than a half or whatever in order for your simulations not to give oscillations. We have the theory of mathematics for those simple problems and for numerics for those simple problems. We do not have the mathematical theory for these complicated problems in biology. We do not have the numerical theory for these highly complicated problems in biology. And that, to me, is really, really exciting, because that means there's lots of open problems to look at, lots of challenges to do. And one thing that's noticed particularly being around here is a number of young people that are here, and so these are the challenges in this field. The mathematical and the computational challenges, just some of them that I think would be very, very exciting for you guys to go and analyze. So what I've done in conclusion, the neural crest showing how simple models combined with experiments can give you new insights. The angiogenesis, how relatively simple models lead to new mathematical challenges. Epithelial sheets, comparison of different computational models. How do we even compare different models with different parameters, etc., is an issue. So I'd like to end there by, first of all, thanking the collaborators who worked on this, the funding, including the funding here that Samara got, and also to thank all my collaborators over the years in the work they've been doing, and again to thank the SMB, and thank you for your attention.
Well, thank you, Philip. So we have time for 4 or 5 questions. So remember that we're recording the talks for the SMB YouTube channel, and the intention is that this serves as an outreach activity for those who couldn't attend to the meeting. They would be able to watch the talks online. So if you want to make a question, please make over the phone.
I had a question about something that you said the very beginning. When you work with biological collaborators, you got so excited that different mathematical methods give you different results because that's something they see in biology. But do you worry that the biologists could have the same problem, that their different results could be because of a technical issue rather than something that is actually
Yeah, so the first thing was that obviously that was a flippant remark. But the second thing is that thing to remember about biological systems is that one thing that characterizes a biological system is that it adapts. And therefore, it is absolutely, should be no surprise at all that the same experiment done in different labs will give you different results. And in fact, we work with people who work on the gut. They have to do the experiment at the same time every day because of the circadian rhythm. So there's so many variables that even once you exclude the fraud that happens in biology, the incompetence, which happens in all areas of, mathematics. Go to a mathematical paper and try and reproduce the computational results. It's the same problem. Once you've excluded all of those, there's still intrinsically these things, the results in the biologists get that are different from each other, they could equally be correct. Either slightly different technique or done at a slightly different time of day or done with a different cell line. Yeah.
Philip, I really appreciated your emphasis on the workflow of model development. As you point out, there are a lot of young people in this room. I would guess that 90% 95+% of them have never developed a model properly in the form of developing a formal verbal model, writing it down formally, and then going through the stepwise workflow to go from a verbal model to a computational instantiation that allows you to do the kind of thing you did in this chaste comparison. If you don't start out by writing a formal verbal model, it's impossible to compare results between methods.
That's right.
And almost everyone in this room does that.
Yeah.
And almost everyone in this room has been taught to do it wrong. And so I think you're really doing a service in showing the right way to build models.
Right.
And maybe there's some way we can get that message out even more.
And thanks, James. I think also that's very important because as mathematicians, we're trained to write down under this assumption, blah, 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 blah. And of course, experimentally, is going back to Kelsey's question, that experimentally, people do write down the methods section. And I guess that's their assumption. But then the problem is that at the end of that paper, if you then say, so now such and such a cell proliferates more than such and such other cell. No, you have to say the assumption, under the assumption of cell type this in fetal calf serum modeled on this day by me, the density increased. It's like us proving the theorem that some function has a 0, and then our conclusion says all functions have zeros. You always have to make sure to read the conclusion based on what the assumptions were. Absolutely agree with that.
Great talk. I appreciate all the work that you've done over the years. So I want to go back to this question of biological heterogeneity because I think it's a extraordinarily important point, and the current phraseology is the crisis of reproducibility, which to a certain degree reflects this degree of heterogeneity that can arise from relatively subtle changes in experimental conditions. Yeah. So I think there's an inherent disconnect between what constitutes a good biological experiment in which you control all the factors and what the intent of math is, which is to increase the generalizability of any knowledge that you happen to have. And I would make the argument that if you focus on reproducing data generated from a highly constrained and highly controlled experiment, you're only examining a very, very narrow slice of the actual behavior space of the model that you're actually the system you're looking at. And you're essentially throwing away a great deal of the heterogeneity that produces the richness of behavior that we see, right? So at some level, what we want to be able to do is encourage our biological colleagues to embrace the messiness associated with different measurements that occur with slightly different experimental conditions. So personally, I'm very interested in the outliers that they have from experimental data, right? Because the natural tendency of a biologist kind of figures something went wrong and throw that away. But barring any sort of actual measurement error, the fact of the matter is that that biological object that you experimented on was able to produce that measurement. And therefore, a computational or mathematical representation of that system should similarly be able to at least reach that particular output in some way. So I think that as we move forward in thinking about how we interface mathematical and computational models with experiment, there's this idea of capturing the broad range of heterogeneity that's seen in these different experimental conditions that would greatly increase the explanatory power of the models that are able to do that.
Yeah, I absolutely agree. And I think that in many cases when a biologist, when somebody says the experiment doesn't work, I actually don't understand what that means. I mean, it mightn't work because you tripped over the Petri dish or something, or you blew up your lab or something. But it might, but if the experiment didn't work because the mice all died, maybe that is the result of the experiment. And so when you say the experiment doesn't work, how much bias are you putting in that? The experiment didn't work because it didn't give the result I thought it would give. So I think that that's very important. In fact, one of the examples I skipped over of when we did this iteration with the biologist, there was a very nice example of that, where they showed us a behavior outcome of experiment which showed invasion, and our experiment showed there shouldn't be invasion. And then they said to us, well, actually, in most of the experiments, we didn't get invasion, but we didn't want to show you a blank screen. So I absolutely agree with you. But that also means that you're going back to what James said about the young people in the room, you've really got to know what the biologists are doing and really work with them and realize, if they're throwing stuff away. Yeah.
Well, with further ado, so I think we have to thank again Philip. So we have coffee break now. Thank you very much, Philip.
Thank you.